Castor, the scientific name is Ricinus communis, belongs to family Euphorbiaceae. Chromosome number is 20. It's a monoecious in nature, cross-pollinated. Castor is an ancient and important non-edible oilseed crop. The genus Ricinus is derived from the Latin term meaning dog tick. It consisting 50 to 55% oil content. Seed and oil have a number of uses. Other names in Bengali it is called as Varando, Gujarati, Divel, in Hindi, Arandi, Kannada, Harlanne, Malayalam, Chittimanku, Marathi, Errand, Oriya, Jadu, and in Telugu, Avadam or Amudallu. Classification: Kingdom is Plantae, phylum Magnoliophyta, class Magnoliopsida, order Malpighiales, family Euphorbiaceae, subfamily Acalyphoideae, tribe Acalyphia, subtribe Ricinae, genus Ricinus, and species Cuminis. Origin of castor is indigenous to Eastern Africa, especially Ethiopia. The origin is Ethiopia. According to Decandela 1890, the origin is tropical Africa. However, according to Wavilo in 1949, Ethiopia is the center of origin. Importance of castor oil. It is important industrial oilseed crop. The dehydrated castor oil is used in paints and varnishes because of its non-yellowing quality. It's a basic ingredient in the production of resins and fibers. Castor is an ideal candidate for production of high-value industrial oil feedstock because of very high oil content, maybe from 48 to 60% of the seed, and this plant is it has unique ability to produce oil with extremely high value, that is about 80 to 90% of ricinoleic acid. Botanical description of castor: it's a plant, tall, glabrous, annual or perennial shrub, grows up to 3 to 8 meters. Plant have developed a taproot system. The stem is erect, partially hollow, smooth and glaucous. The leaves are usually a glossy green with prominent veins or on undersurface. Inflorescence. Inflorescence is called as raceme. This is borne terminally on main and lateral branches. It is normally monoecious plant, about 50 to 70% male flowers occurring towards the base of racemose and 30 to 50% female flowers in upper portion. That means female flowers placed in the upper portion and male at the basal portion. Female flowers. Flower buds are elongated and narrow conical in shape. These are female flowers, red color in appearance. It has perianth and ovary is comprised of 3 locules. Usually each of the 3 carpels has a single ovule. There are 3 fringed and sessile stigmas with bright color. After the fertilization, the ends of stigma dry up, pistillate flowers being borne in the upper part of a raceme open after the male flower open. About male flower. The buds of the male flower are flattened, conical in shape. Flower is regular and hypogynous. It has 5 segments of the perianth and has many stamens united into several bundles, commonly called polydelphous. The pollen grains are small, lightweight and oval in shape. These are the pollen grains. After flowering, pollens are dehisced for 1 to 2 days. Historical background of castor. Exploitation of hybrid vigor in castor was taken up quite early in 1950s in USA and USSR. Increase in yield in the experiments conducted between 1951 to 54 by Moshkinin and during 1986. Weibel and Woodworth in 1946 suggested the use of natural crossing plots for the production of hybrids in Illinois. The method for pollination control for hybrid seed production was developed 4 decades ago. That was after 1950s. Recent reports indicated that presence of substantial amount of heterosis for yield and its components. Development of hybrids. Research work in the India gained momentum during late 60s with the initiation of All India Coordinated Research Project on Oilseeds under ICAR. First Castor hybrid GCH3, that is a cross between TSP 10R into JI15, was released in 1968-69, which yielded 124% higher than S20. The development of pistillate line VP1 based on TSP 10R gave new impetus to hybrid development program in the country. India is the first country in the world to exploit hybrid vigor in castor. The hybrid development is based on the pistillate line used as female parent. Therefore, development of pistillate line is very important in castor. Development of pistillate lines. Let us see how to develop them. Castor is known as sexual polymorphism, that is, there are a number of sex variants found in one or the other genetic base and seasonal conditions or growth stages. Among these variants, 3 types of femaleness is observed, that is NS and NES type, used in hybrid seed production. First is N type. A recessive sex switching gene, that is recessive F, governs femaleness. Inbred pistillate line of this type designed as N145-4 by Casson and Honaf. Homozygous recessive plants are gynoecious and heterozygous plants are normal monoecious. This system can be maintained by sib mating and seeds from female plants are harvested separately. S type. This system is based on sex reversal variants which behave like polygenic complex with dominant and epistatic effects. Sex reversals are plant variants which start out as female and then revert to normal monoecious. Development of stable pistillate S type line is based on selection from order revertents. The late revertents produce more than 90% pistillate plants which become highly suitable for hybrid seed production. NES type, this is third type of pistillate condition which is homozygous for N pistillate gene, that is small ff, and also contains environmental sensitive genes for staminate interspersed flowers. NES type of pistillate line can be maintained during summer whereas hybrid seed production can be safely taken up during normal rainy season planting. Senes 1, developed by Zimmerman and Smith during 1966 and JP65 by Enkinidu and Rao in 1973, are NES type of lines. Sex expression in castor. It's highly cross-pollinated crop and has highly polymorphic sex expression. Natural outcrossing as emasculated that estimated range between 70 to 90%. Female tendency is in this crop is higher under long day length and cool dry conditions. Based on elaborate studies, it could be categorized into 2 of genes for sex expression. The first group expressed of qualitative genes which determines the kind of flowers in the inflorescence. The second group, comprising polygenes, was postulated to be responsible for gradient differentiation and racial differences in sex tendency. Genetic resources or where these are stored. A Search of international germplasm collections on biodiversity website combined with USDA ARS Castor germplasm at Griffin, USA identified 12 major sources of germplasm. Total of around 6,600 accessions, extensively germplasm collections are held in Brazil, China, Ethiopia, India, Kenya and USSR. Hybrid seed production in castor. Hybrid seed production, unlike most other crops, in castor is a highly skilled and specialized process because of the typical breeding system complexity of sex mechanism. Its high sensitivity to genotype into environmental interaction. Steps: 2 steps: maintenance of parental line, that is foundation seed production, and second is production of hybrid seed, that is certified seed production. Isolation. Minimum isolation distance of 150 meters should be maintained for certified seed production. For foundation seed production, 300-meter distance is required. Planting ratio mandatory is 3:1, 3 is female and 1 is male, followed in hybrid seed production plots. Planting time. Choice of season and planting time have significant bearing on yield and seed quality and ultimate returns from hybrid seed production plots. Any further delay or advancement in planting beyond the recommended time schedule will have undesirable effects on sex expression of the female parent. Different female parent for different seed types. Female parent in southern or western zone, for nucleus and breeder seed, the female parent should be planted during second fortnight of January in western and northern region, in second fortnight of February. Foundation seed in southern zone, the January first fortnight, and in western and northern zone, first fortnight of July. Varieties and male parent, for nucleus or breeder seed, the planting in southern zone is January second fortnight, and in western and northern it is January first fortnight. Foundation seed, in southern zone it is June second fortnight, and in western and northern it is July first fortnight. For hybrid seed production, certified seed, in southern it is September second fortnight, and in western and northern it is August second fortnight. Some popular hybrids and their parentage and released years are given. GCH3 was the first hybrid in India. The parents are TSP 1 OR into JI-15, released in 1970 by Gujarat Agriculture University. Then Gauch 1 was a cross between VP1 into VI9, released during 1973. GCH2 was a cross between VP1 into JI35, released during 1985. Then GCH4 was a cross between VP1 into 48-1 during 1986. Deepak was a cross between DPC9 into DCS9, released during 2001 for the regions of Andhra Pradesh, Tamil Nadu, Karnataka and Maharashtra. Major constraints in exploiting heterosis in castor. The one reason is stability of pistillate line, creating problem in hybrid seed production. Second is wilt under irrigated condition and root rot under rain-fed condition. Lack of early and high-yielding hybrids or composites for rain-fed area. Lack of knowledge of hybrid seed production technology among seed producers, resulting in large-scale rejection of foundation and hybrid seed plots. Thank you. Hope you liked this video, and do not forget to subscribe my channel Varsha Gaitonde. Thanks once again.